You know how many people normally watch the streaming on the YouTube?
How many people, sorry?
Do you get many people watching the streamed seminars on YouTube?
It I mean, depends on the day, but yeah, we have maybe less than 100, but depending on the speaker. But yeah, sometimes problem is that with the Zoom we don't have enough space sometimes, and if we reach 100 people, then they cannot get into the seminar. So that's why we do it this way. So
okay, that makes sense.
We're gonna start. Let me just make sure. So good afternoon, everyone. Today I have the honor to introduce our speaker, Dr. Jonathan Houseley. He got his master's in research in molecular functions in disease at the University of Glasgow, where he also got his PhD, in working in Drosophila as a model of myotonic dystrophy. Later, he joined the lab of Professor David Tollerby at the University of Edinburgh to start his postdoctoral research, where he worked with the initial in vivo characterization of the TRAMP model and its roles in RNA surveillance. Later, in the same lab as a Leverhulme Trust Early Career Fellow, he described non-coding transcripts targeted by the TRAMP complex at telomeres and in the ribosomal DNA, and showed that the TRAP complex is required for the stability of ribosomal DNA. In addition, he discovered and characterized a non-coding transcript in the GAL cluster, which remains one of the best characterized antisense non-coding RNA systems. In 2009, he started his group at the Babraham Institute in Cambridge, and he started as a Wellcome Trust Career Development Fellow, then became a senior group leader, and he's currently a Wellcome Trust Senior Research Fellow. His work has been published in high-impact journals, including eLife Plus Biology, PNAS, Nucleic Acid Research, and Cell, and he's studying the mechanisms that lead to genetic and epigenetic change. Particularly, he's interested in the ability of cells to alter their heritable genomes in response to particular environmental conditions, and therefore to rapidly adapt to changing environments. His group is starting to discover connections between adaptation and aging, and today he's going to share some of their results. Welcome, John, and thanks for accepting our invitation.
Thank you very much for inviting me, Paula, and also thank you to Myra. I don't know if you're watching. So I'll just share my screen. Okay, we all good? So today I'm going to talk about aging. So my lab has been doing aging research for quite a few years now, and it's sort of dawned on me that I very rarely, if ever, actually give seminars about it, because what we do doesn't ever seem to sort of form into a very coherent story. And I think that will probably be quite obvious in the seminar. And also the concepts that we have ended up working with are difficult, and so can be quite difficult to present. So I hope we'll end up with sort of a clear story and some take-home points. So, basic question that we're interested in, like everyone who works on aging, is why we age. And this is an important question, because if we're going to actually do anything about aging in terms pharmacologically or in terms of lifestyle, we sort of need to actually understand the process at the molecular level. And the fact that we don't really understand aging, I think, is a problem. And it's difficult to grasp this idea that we don't really understand aging, because culturally we get it. Everyone understands aging. It just seems like a natural process because everything around us wears out, everything around us ages, and we're sort of comfortable with the idea that actually just aging is an inevitable consequence of biological life. When I started reading about aging, 7 or 8 years ago now, I got really caught up with the problem of lifespan. And this is a problem that many of you will be aware of, which is that lifespans don't make sense, because you would think if aging is a basic result of biological life, then organisms that are closely related should have similar lifespans, but they don't. So for example, if we look at mammals, we've got a 2 order of magnitude difference between our mouse and our blue whale. Now, obviously, it's a bowhead whale. So obviously the mouse and the bowhead whale have very different lifestyles, and one lives in the water, etc. So that could be a big difference. But even within rodents, here we've got a order of magnitude difference in lifespan. Now, still, the squirrel's quite a lot bigger than mouse. But if you compare the mouse to this bat, it's about the same size, yet the bat lives for over 40 years. And it's really hard to understand this on any sort of model of aging which involves some sort of accumulation of damage. And particularly if you start thinking about this in terms of cells and molecules, it's really hard. Because if you think about, say, a neuron in this mouse and in the whale, they're both mammals. The neurons are pretty similar to, you would have to be, if you look to them in isolation, you'd need to be a fairly skilled cell physiologist to tell the difference. Yet the mouse neurons are turning into a neurodegenerated mess in a few years, while the bowhead whale neurons, they're still developing. So there's just this really difficult problem that there doesn't seem to be any connection between what an organism does and how it lives and the aging process. And as I was reading about aging, I kept coming across these ideas that actually aging might not be just something that comes about because of damage, but it might actually be an evolved, useful process. And these ideas are definitely around on the fringes of aging research, because the problem is they're really hard to justify. They've been kicking around for a century or more, but no one's really got to the bottom of this. And one particular problem is, if you have an evolved process, it tends to be evolved for a purpose. So you've got to imagine that you should be able to find some sort of fitness advantage this is a result of a particular process. But actually, as far as we know, an organism that ages has less chances to reproduce than an equivalent organism that doesn't age. So aging reduces reproductive fitness. Aging, therefore, almost by definition, can't be under positive selection. And you're left with what is essentially a null hypothesis, which is that aging isn't some sort of inevitable consequence of some sort of damage that gets accrued during life. When we started thinking about this in my lab, we sort of thought that we might be thinking about this in the wrong way. Because, as I said at the start, aging occurs in pretty much every living organism that we know about. And so if it is some sort of evolved process, then it didn't evolve for the benefit of the palm tree or for the benefit of the zebra. It must have evolved for the benefit of the last eukaryotic common ancestor, and that was probably a unicellular organism. So we started asking the question, well, unicellular organisms have very different lifestyles to multicellular organisms. Can we identify reasons why aging might be useful to a unicellular organism? And then we could maybe imagine that if it evolved in our last eukaryotic common ancestor, it may have become such an important part of being a eukaryotic cell that the aging process actually simply was inherited from this last eukaryotic common ancestor and is still with us today, not because it's useful to us, but because it was useful to them. So what I'm going to really focus my talk around today is that basic question of what could possibly be useful about the aging process, because this is really the problem with any evolution-based argument about aging. We have to be able to say, here are some absolutely clear conditions in which old cells have a fitness advantage over young cells. And of course, at the moment, we don't really have any situations like that, because aging seems to be just across the board, process of decline. And so we started by making a fairly outlandish hypothesis, which is that what aging might be useful for is promoting adaptation at metabolic and genetic levels. And the reason that we made this hypothesis is that my lab at the time, and still, works on adaptation at metabolic and genetic levels. So we just asked whether we could slot aging into the research portfolio that we had. So our unicellular model organism of choice for looking at aging is budding yeast, Saccharomyces cerevisiae. It's a very well described model organism for aging, although aging works a little differently in cerevisiae than it does in higher eukaryotes. So we measure age not in terms of time, but rather in terms of numbers of cell divisions. So each time a budding yeast cell divides, it divides into a mother and a daughter, and the mother can only undergo a finite number of divisions before it can no longer divide. What's called, it reaches the end of its replicative lifespan. The daughter cells, on the other hand, have their replicative lifespan reset to 0. So each daughter cell starts this process again. When this one divides, it will become a mother, and its lifespan will increase, and it will have the same lifespan that the mother did, or thereabouts. And we've known for a long time that as mother cells age, they accumulate oxidative damage to proteins and genetic changes of various sorts, and basically lots of things that seem to be associated with aging, and the aging process responds to TOR signaling and caloric restriction. So basically it feels like a similar aging process to that which we see in higher organisms. And so we can define some particular pathologies that accompany the aging process. So the first is the onset of senescence. This is a word that's rather unhelpfully used in the yeast literature and in the mammalian literature to describe processes which are almost certainly not the same process. Rather, they just describe the pathology that happens at the end of life. So close to the end of a budding yeast lifespan, its cell cycle starts to slow down. The cell cycle timing becomes more heterogeneous, and it starts to show obvious pathologies. So this one that you'll be seeing a lot in this talk is aggregation of mitochondria. So in young yeast cells, mitochondria form these little strings. But in old yeast cells, these form into these massive autofluorescent blobs, which we can measure very easily and quantify using imaging flow cytometry. And as the cells increase in age, basically the intensity of the GFP signal we see off this mitochondrial marker just increases in a pretty linear way. So it's a very useful way of monitoring the onset of aging pathology. Also, it's been known for a long time, long before our work, that there is a transcriptional change that accompanies aging in yeast. So what I'm showing you here in a very squished up form is the expression of every single gene in the yeast genome as the cells age. And you will see that pretty much every gene is increasing in expression, and pretty much every gene is changing in expression. This makes for fun normalization issues when we're trying to actually analyze the data, and most of them increase. And we can represent this in what's called an MA plot. So in an MA plot, we're comparing two conditions, old and young cells, and we're plotting each gene on the x-axis being the average expression of that gene. And on the y-axis, we have the change in expression across the conditions. So ones up here increase, genes up here increase in expression with age, genes down here decrease in expression with age. And you can basically see that almost a whole transcriptome is up in this half. And this is known as a genome-wide loss of transcriptional regulation. No one understands why it happens, but it seems to basically be some sort of epigenetic breakdown that happens in these cells. So we have things that we can measure and that give us a readout of how the aging process is proceeding in these cells. And so we started out quite a while ago asking this very simple question: can we find a fitness advantage that comes from aging in yeast. This doesn't necessarily have to be to the individual cell, but it certainly needs to be to its direct descendants. We didn't want to play with kin selection or any of these other ideas, because to be honest, we're not an evolutionary biology lab, and beyond this simple evolutionary biology, we're out of our comfort zone. But what we thought was that when people talk about cells getting less fit or yeast cells getting less fit, the experiments are almost always done on glucose, because glucose is yeast's favorite carbon source. In fact, budding yeast are what's known as a specialist organism. Even if you present them with a whole smorgasbord of different carbon sources, they will consume the glucose and nothing else. And what we asked was whether, if you, as the cells age, although they lose fitness for glucose, do they lose fitness on the other carbon sources, which they can live on, they're just not particularly happy about it? Or actually, do they gain fitness on these other carbon sources? So rather than just a loss of fitness, this would be a transition in lifestyle from a specialist organism that focuses on one carbon source to a generalist that focuses on whatever it happens to have to hand. So my PhD student at the time, Stephen Frank, set up a really subtle, very simple assay for looking at this, where what he would do, he had two cultures of cells growing on glucose, which carried a differential marker. We had a culture of young and aged cells, and he would mix them together. He would inoculate them in an outgrowth culture with different carbon sources. He would let them outgrow for 10, 15 generations, and then he would look at the markers that were in these to work out how many of the cells in this culture came from the young and how many came from the old. So it's a classic competition assay. And if you do this experiment and inoculate the cells into glucose, what you see is a very small advantage for the young cells. And that's at first sight seems quite surprising, but it's what we expected. Because of course, these competitions take place over 10 to 15 generations. But as soon as the aged cells have undergone one division, they have produced a new population of young cells. So actually, the only thing that's different is the first cell division, and then the cells should grow at roughly equal speeds. So what this represents is the difference in fitness of the aged versus the young cells for that one division. And that point about it being a quite small change really made what we saw in the other cultures incredibly surprising. Because actually, when we did this experiment in galactose or raffinose or glucose with ethanol added to it, we saw that the aged cells massively outcompeted the young cells. So in galactose, which gave the strongest effect, 80% of the cells in the outgrowth culture came from the aged progeny, and whereas that was inoculated with a 50-50 mixture. So across 10 to 15 generations, this is a massive, massive selective advantage that the aged cells appear to have. This is really surprising, but it was the first evidence we had that there is a proper selective advantage for aging. And the more we thought about this, the more we thought, well, actually, this sort of makes sense, right? Because yeast specialize on glucose, we know this, but they're a colonizing organism. So a cell will colonize this nice glucose and fructose-rich bunch of grapes. But then being a yeast, what they do is they eat their environment and they turn it into ethanol. So after some amount of time, what's left is a whole bunch of carbon sources which are not glucose and some fairly toxic ethanol. And at that point, the specialist young yeast have nothing they can really do. They're not adapted to this situation, so they're going to stop growing. But the old cells, which do better in other carbon sources and better tolerate the presence of ethanol, will actually be better able to grow on these other carbon sources. So basically, what the most successful cells will do is maintain a specialist mode of growth until the glucose runs out. And then simultaneously with the glucose running out, they need to switch their metabolism. So they need to make a prediction about how long it's going to be before the glucose runs out. And that's sort of what aging does, actually, because it basically says that the more cell divisions you have undergone in the glucose-rich environment, the more likely it is it's going to run out in the near future. So what the cells are trying to do, what cells seem to do with this process, is basically take a bet on when the glucose is going to run out, and they're just slowly but surely shifting their metabolism in the right direction, that they'll be able to outcompete any of the specialists when the glucose runs out. So that, of course, makes another prediction, because that suggests that this is all about glucose and fructose, which is metabolized in a very similar way. So we shouldn't see this on other carbon sources, and that's a very testable prediction. So we did some equivalent experiments on galactose, which gave the strongest effect in the competition assay. So here, we're not in any way restricting the diet. We know that dietary restriction, caloric restriction has an effect on the aging process. We're not doing this here. We're just changing the cell's diet, but we're giving them as much galactose as they want to use. But now there's no advantage to this metabolic switch. So if the aging process is all about this metabolic switch, these cells are already growing on their non-preferred carbon source, so they shouldn't change. And indeed, what we found was that although the lifespan of these cells is very similar, the senescence mark was almost undetectable in the cells aged on galactose compared to those aged on glucose. And similarly, we did some cell cycle analysis. These are horrid experiments that very brave research assistant in my lab, Grazia, undertook. But basically what she did was she aged some cells on glucose or galactose, and she measured the cell division time of individual cells. And when the cells were aged on glucose, not a huge amount of slowdown was observed. And this is not surprising because it was really hard to do this experiment. So actually these cells, they're only at about half their maximum lifespan at this point. Nonetheless, she started to see that the cell cycle time became more heterogeneous, which is what people have observed in microfluidics experiments. But when she looked at cells aging on galactose, she saw exactly the opposite. So the cell cycle time got a little bit faster, which was quite surprising. And even more surprisingly, it got much more precise. So actually, as these cells age on galactose, they seem, if anything, to be getting fitter rather than less fit. And we looked at the transcriptome. So again, I'm showing you this transcriptional deregulation that you see when you age cells in glucose. But when we look at these in galactose and at age-matched cells, we see almost nothing. So the vast majority of genes are clustered around the center line, which means that they're not, there's no global change in gene expression. So and we did this experiment on various diets, and every time we found that only on yeast preferred carbon sources, glucose or fructose, do we see aging pathology, and not on anything else. And then another of my PhD students, Dori, then went on to ask, well, when does diet matter for these cells? Because suddenly this aging process seems to be really dependent on diet. So she did another conceptually simple experiment. She aged cells to middle age on glucose or galactose, and then she spun the cells down, and she then re-inoculated them into glucose or galactose and let them grow for the rest of their lives. So what we separate is an early and a late life feeding. And if you look at the cells aged on glucose for 24 and 48 hours, just looking at the senescence marker here, we can see that the senescence marker accumulates as before. And the galactose one goes from here to here to here. So basically there's no change, real change across this experiment on galactose. And again, that's what you said before. What's interesting, however, is that the cells that started life on glucose, but then spent the late part on galactose, show almost the same amount of our senescence mark as the cells that were grown all the time on galactose. Whereas the cells that were grown for the first 24 hours on galactose, they do not accumulate the senescence mark, even if they spend the later part of their life on glucose. So what this tells us is that the important time to define how well these cells age, whether they accumulate these markers of pathology, is not what's happening to them in late life, it's what's happened to them in early life. It's a rather depressing conclusion. But this led us to ask, what is actually so special about glucose? So we know that whatever happens, whatever it is that's important, it happens in early life. So we stared at the transcriptome data that we had from these experiments, and we asked, well, what's different? We did a very simple differential gene expression analysis between cells that were young or old, growing on glucose or galactose, and then we did a Go analysis of what was different. Obviously, there's lots of things that are different because cells growing on galactose, they have a whole different set of metabolizing genes. But the thing that was really obvious was that the Go categories that were most enriched were involved in extraction of energy, again, not too surprising that, and particularly respiration. So basically what is driving these Go categories is this one, respiration. So basically it's expression of nuclear mitochondria genes. That was sort of awkward and sent us scurrying back to the textbooks because again, respiration is not really something that our lab know a great deal about. But textbooks have been well written on this sort of area, on this sort of topic. And so we have a very good understanding of how this works. And you'll be very familiar with this. So what basically happens is you have your carbon source goes in here, for example, glucose. This is broken down to pyruvate, and under aerobic conditions, the pyruvate goes into the mitochondria, where it's broken down by the TCA cycle to generate energy. On the other hand, if there's no oxygen around, this goes into a fermentation pathway. However, this is what happens in most organisms, but yeast being yeast is different because actually they have something called the Crabtree effect, which means that when you put them on large amounts of glucose, even if they are fantastically oxygenated, they don't use their mitochondrial, they don't use oxidative phosphorylation really at all, but rather they push everything into this pathway, which results in ethanol, which is good if you're making beer, and results in carbon dioxide, which is good if you're making bread. But seems to be not so good if you're talking about aging. Whereas if yeast are grown on galactose, then they have something rather more subtle. They have a mixed metabolism. So galactose is processed into fructose phosphate as the first step of its metabolism. Then it goes down the same glycolysis pathway, but actually it goes into, I would say, fairly poorly defined mixture of fermentation and oxidative phosphorylation. The important point for this story being that yeast use their mitochondria on galactose. My slide is wrong here. Yeast use their mitochondria when they're growing on galactose, but they don't use their mitochondria when they're growing on glucose. So this raised a fairly obvious hypothesis that that the aging pathology is all about respiration. We thought, well, we can test that. So we can firstly just prevent the cells growing on galactose from respiring, and we can do that in a fairly blunt way by knocking out a key component of the electron transport chain. And when we do that, now the cells start to accumulate our senescence marker. So now we get aging pathology in galactose. And conversely, if we try to boost respiration in glucose, can we suppress the pathology? This is really hard to do actually, because yeast are really hardwired to undergo fermentation on glucose. But we can at least drive mitochondrial biogenesis, which forces a bit of mitochondrial function. And we find that this significantly reduces the appearance of our senescence marker, although eventually it catches up because the cells do continue to ferment. So we can reduce aging pathology in glucose by driving cells to respire. And if we prevent the cells growing in galactose from respiring, they start to show the aging pathology. So, in conclusions for this part, what I've shown you is that aging seems to improve the adaptability of cells. This only makes sense in terms of their life history strategies for glucose, and actually the aging pathology is specific to glucose and results from an early life suppression of respiration that happens in glucose. So what this basically means is that the aging pathology doesn't seem to be part and parcel of the aging process. Remember, I said that these cells growing on glucose and galactose have about the same lifespan, but the ones that are growing on glucose have pathology, and the ones that are growing on galactose don't, as far as we can see. And so this whole aging pathology seems to be something that the cells actively control because it's useful and makes sense in their life history strategy, because they start out as young cells in glucose with maybe some other stuff around, which they ignore. But over time, that is converted to ethanol and some other stuff, which now the cells want to metabolize. And these aged cells, by the process of becoming aged and through the process of fermentation, now are better able to metabolize what's left. So this is how and this is one way in which aging pathology, or at least aging pathology as it appears to us, can actually be useful to an organism. So the second part of my talk is really to try and link this through to genetic change, which is the other thing my lab is really interested in terms of adaptation. And there's sort of rather tenuous link between these two parts is that it is known that yeast genomes do change with age, and they change a lot. So this is from some genome resequencing experiments that we did, where I'm looking at just the genome size. So basically the number of base pairs of genome in each cell. And what you see is that most of the genome in black doesn't change very much, but the ribosomal DNA in red is getting much more abundant as the cells age, which is weird. So just to put this in context, basically what we're talking about is maybe a 1.5 times increase in the whole size of the genome that occurs in these cells as they age, which is being driven by an accumulation of ribosomal DNA. And it's been known for a long time that that is not ribosomal DNA that's accumulating on the chromosome, but rather it's accumulating in the form of circular DNAs containing ribosomal genes. So basically, think of them as plasmids. So these basically huge numbers of plasmids accumulating in the nucleus of each cell. And these have been known, again, for a long time. So they were described a quarter of a century ago by David Sinclair and Lenny Garanti, with this incredibly influential Cell paper where they described the formation of these circles, which said are massively abundant, and speculated not unreasonably that they may well be what's causing aging in yeast. And the reason that these become so abundant in age is because of the way that they're handled during cell division. So each time a cell, so at some point, presumably in a fairly young cell, we get one circle formed by some random recombination event. When the cells divide, these circles are replicated because they've got replication origins in them. But what they don't have is a centromere, and yeast decide how to segregate the DNA in their nucleus based on whether it's got a centromere or not. Yeah. This comes down to basic mechanisms of mitosis. And actually what yeast do is identify acentric DNA, so lacking a centromere, and they retain it in the mother cell. So now, as the cell divides, it's replicated this DNA, but it's kept it in the mother cell. So now the copy number of this circle has doubled. as the cell goes through another division, again, these replicate, and again, they're retained. So the copy number of these things increases exponentially with age. And this massive change has been long suspected to drive aging. It's really hard to prove. So the data is very correlative, but there is a lot of data there. But this sort of left us scratching our heads a bit, because there's a mechanism which retains these circles in the mother cells. This has been worked out by Yves Barral's lab. It's a very nice mechanism. And it revolves around the fact that yeast have a closed nuclear division. So their nuclear envelope doesn't break down during the cell division. And the nuclear pores that are in the mother cell stay in the mother cell. They're not passed on to the daughter. And so these complexes, Saga and Trex2, bind to these non-centromeric DNAs, and they attach them to the nuclear pore complexes. And that leads to this almost perfect retention of these circles in the mother cell nucleus. And therefore, this leads to the pathology, which left us with a question. Why the hell would they do that? If these are such bad, damaging molecules, when you accumulate them, why on earth would the cells accumulate them in this way? And it left us thinking, well, maybe actually there's a reason for doing this. Maybe there's a reason for retaining circles, not necessarily the ribosomal DNA circles. As far as we've been able to tell, we can't find anything that those do. But there's plenty of other circles. And we thought that this might help us with something that a problem that we've been wrestling with for a long time in the lab, which is the basic idea of how adaptive mutations arise. Because adaptive mutations are difficult to find for any organism. And I talk about this in a way that sort of assumes that the cells have some control over this. Of course they don't, and I'm anthropomorphizing, but it's a useful way to think about it. But basically, finding a useful mutation is a real problem, because most mutations aren't useful. A lot of mutations do nothing, and quite a lot of mutations are actively damaging. And the proportion of mutations that are actually useful in any particular situation is vanishingly small. But you've got to have mutations, right? So you've got to have some level of mutation rate in any organism, otherwise they can't evolve, because mutation is the substrate for evolution. But if your mutation rate is too high, you have too many, you have an accumulation of negative mutations. And this is a real problem. So you can sort of imagine how this works, right? So you have a population of cells that's expanding up this way. At some point, a mutation occurs, which like many mutations is a bit damaging, and so this cell is doing less well than the progeny of the rest of the population. And then certain mutations also make cells incompatible in terms of mating. So chromosomal rearrangements would be the classic example. And so as the cells accumulate more mutations, even if they're neutral, what you tend to have is more mating incompatibility. Mating tends to be the thing that deals with difficult mutations and allows them to be repaired. And mutations start, if you have a high mutation rate, you'll have mutations appearing on an already mutated background. So even if this useful mutation appears here, it may well be appearing in a background which is already mutated. So the way this is classically seen to work is that your mutation rate needs to be balanced such that natural selection is removing any damaging mutations faster than they're forming, such that when a new and useful mutation arises, it's already arising on a clean background. And that's all very well, but it's slow. And unicellular organisms particularly are at the mercy of their environment. They need to be able to adapt quickly. And so relying on this sort of process is really problematic. But circular DNAs might provide an answer to this. So going back to this idea of these acentric circular DNAs, these circular DNAs, if they accumulate, what they're doing is they're altering the genotype of the cell without altering the chromosomal genome. And if those happen to contain genes, then the copy number of those genes will change. That can have phenotypic consequences. And we know this because there's a whole field of research that is looking at this in cancer, and it's only a big thing in cancer, that actually has been realized that the oncogenes and drug resistance genes that are amplified up to very high copy number in cancer cells are very often present not on the chromosomal DNA, but on the circular DNAs. And the reason for that is that these circular DNAs are very unstable. They can be amplified and contracted in terms of copy number very easily. And this means that the cells can adapt far more quickly than they would by mutational processes that affect the chromosomal DNA. And so what the circular DNA might allow is the formation of a temporary but reversible genetic change. So you form a circle in a young cell. That doesn't change the parental genome. An old cell might accumulate quite a lot of copies of some circles that happen to be present when it was a young cell. And if those circles happen to contain genes, and obviously if you excise random bits of the yeast genome, you get some genes because it's a pretty compact genome, then that will have phenotypic consequences. And the phenotypic consequences of this are normally bad. There's no two ways about that. If you take a random couple of genes and you amplify them up to 50 copies while all the other genes that normally work in a gene regulatory network remain at the same copy number, then that's almost certainly going to break your genetic network. But once in a while, you might get a situation where that amplification is useful and provides a fitness advantage, particularly in response to some sort of damaging, toxic environmental change that might impair the growth or kill off the rest of the population. So basically what this provides is some genetic heterogeneity, which is in the old cell population. And this led us to start thinking about some really wacky concepts. So the idea that maybe actually these circular DNAs could form basically a bespoke genome that is tailored to the environment, that sits on top of the normal chromosomal genome. And the way we figured this might work is that we know cells can sense the environment and they can activate gene expression processes in response to the environment. Nothing controversial about this idea. And what we wondered was whether the induction of genes in response to the environment made those genes more likely to be occasionally turned into circular DNAs. So this is an idea that's been floating around for quite a while. There's good mechanistic support for it, and we've been able to show that this actually happens, which again, I'm not going to go into now. But basically, it comes down to the idea that a highly expressed gene generates a bit of DNA damage, and that makes it more likely that you'll get recombination events that result in the circular DNA forming. The important part of this concept is that now we have two copies of the gene, one on the chromosome, one on the circular DNA. Now our cells age and the circular DNA goes through this amplification process of being replicated and retained. So now this gene that was induced in response to the environment has been amplified. And if we imagine this happening across a whole population, what we might expect to see is that genes which are being induced in response to the environment more often end up on circular DNAs, or rather, to turn that idea around, a gene that is inducible in response to the environment will more often be in circular DNA and accumulate in the circular DNA fraction when it's being transcribed than when it's silent. So all around the genome, if we've got some sort of background processes that just generate the occasional circular DNA, those will be biased towards genes which are being induced in response to the current environment. So again, we wanted to experimentally test this, and we looked at copper resistance for this. Don't worry too much about why copper resistance. From our point of view, it's just model environmental stress, very, very well defined in yeast. So copper causes stress and cell death at high concentrations. And there is a single resistant gene called CUP1, which encodes a metallothionein, which basically binds to the copper and titrates it away. And so the extent to which cells are resistant to copper does largely depend on the copy number of the CUP1 genes. Basically, more copies allows the cells to make more of this protein and titrate away more copper. And so you can imagine that a cell with a few copies of the CUP1 gene has relatively low resistance to copper, and a cell with more copies of the CUP1 gene have higher resistance to copper. And this has been very well established over many decades ago by other labs. And the important thing to note is that this CUP1 gene is induced when cells detect copper in the environment. Okay, so we have an inducible resistance gene that is induced in response to copper in the environment, and the absolute copy number of that gene in the cells dictates how resistant to copper that cell is. And so our prediction would be that this CUP1 gene might turn up on circular DNA more often when the cells are exposed to copper and it's transcriptionally induced. So Ryan, a PhD student in the lab a couple of years ago, decided to actually test this. So what he did was he grew cells in the presence or absence of copper, and then he used some good old-fashioned radioactive southern blots to actually look for circular DNA within these cells. So here we're using gels that separate the chromosomal DNA here from circles present up here. When he looked for circles in the total population, couldn't see anything. But of course, most of yeast population is young. The age cells are actually very rare. And when he purified out the age cells, then he could detect circles containing this CUP1 gene, and they were more prevalent in the cells that were aged in the presence of copper than those that were aged in the absence of copper. And this wasn't just some artifact that was driven by there being more circles formation in general, because the ribosomal DNA circles were more prevalent in the absence of copper. And then he isolated that DNA and he sequenced it. And I realize I'm running quite short for time here. And what we could then see was that the cells that were growing in the presence of copper accumulating more circular DNA mapping to this CUP1 locus. And when we looked genome-wide, we saw that most of the genome doesn't produce circles. Some of it does, but this is not affected by copper. The only bits that do are these regions up here, and these all come from the CUP1 locus. This is an amazingly locus-specific response, forming these additional circles in response to copper. Now, of course, this could be a selection event. The cells with more CUP1 circles might just survive better, or it could be a gene expression event. So to make sure we were looking at something driven by the gene expression event, we mutated the system. So we took the CUP1 gene, got rid of it, replaced it with a functionless tag. And also, because copper is pretty toxic, nasty stuff to work with for the cells, we also changed the promoter so we wouldn't have to include copper in the system at all. And we swapped this out for a galactose responsive promoter. So what we then have is cells which produce a completely functionless gene in response to an environment which they're perfectly able to deal with, so a non-toxic environment. Nonetheless, when we aged the cells in galactose compared to glucose and repeated this sequencing experiment, what we saw was that in galactose, we saw an increase in the formation of circular DNA coming from the cut1 locus, so these bits and these bits, which we hadn't mutated out, and also from this GAL110 region and the HA gene. So basically, we're getting a very selective, tailored circular set of DNAs. So overall, this fits into quite a bizarre but interesting model. So we know that young cells contain various of these circular DNAs, some of which are driven by the environment, some of which are just occur at random, and they're different in each cell. As these cells age, the circles replicate and they accumulate. And in most situations, that's going to be unhelpful or actively damaging. But just occasionally, it can be useful, right? Because if this blue circle encodes, for example, our CUP1 gene, then these cells should better tolerate an environment that suddenly becomes more copper rich, whereas the other cells, and the young cells in the population, may well die off. And because this is a heterogeneity, this process increases the heterogeneity of the age cell population, you have other cells which might do better when a different environmental change happens. So for example, this yellow circle might encode a gene which helps the cells to survive whatever this yellow environment is. So what this means is that as aging cells accumulate circular DNA, yep, they're accumulating all this ribosomal DNA circles, and that might be bad, but they're also accumulating genetic heterogeneity. And the age cell population ends up being extremely genetically heterogeneous because each one is amplifying a different set of genes, although these are, as I said, slightly biased based on the environment they're in. And as I said, mostly that's bad news. It's going to contribute to the aging phenotype. But also, sometimes it can be a lifesaver. So overall, what I've been telling you today is that there are aging mechanisms that we can imagine, and we can actually see, which are useful to unicellular organisms. I've shown you that aging in yeast is beneficial under their normal way that they grow, and also aging increases their genetic heterogeneity. Does this tell us that aging evolved? No, of course it doesn't. All it does is provide a rationale for ways in which aging could have evolved. And this is important because at the moment, we don't understand what drives the aging process. We don't understand what regulates it. But if we can understand that aging has a purpose in unicellular eukaryotes and there's a logic to it, it should be easier to try and dissect the drivers and the control mechanisms. I'm going to finish there and just thank people who did the work, Steve, Ryan, Dory, or the Wellcome Trust who funded us, and the BBSRC who funded us, and you for your attention. Thank you. I'm also going to turn the light on because it's got dark as I've been talking.
Thank you, Jen. I think we have a question from Gabriel.
Hi, this is Gabriel.
Hi, Gabriel.
So very interesting your work. So I have the following question. By seeing your results, they look very much similar in many different ways to what has been described as an epigenetic memory in yeast. If you use glucose and galactose, you can see that the yeast transcriptionally responds faster when you expose the yeast that were exposed before to galactose or repressed with glucose and then exposed for the first time to galactose. And that has been shown to be related to chromatin changes. So I'm not sure, and this is a matter of the term that you're using, aging in organisms has a different meaning for what you are characterizing in this unicellular organism. And so what I'm wondering is if what you are studying is actually memory or some kind of memory rather than aging. So I'd like to hear your opinion because you were saying that one of your main points was to convince yourself and the rest that aging is something that is common to unicellular and multicellular organisms, and consequently you could find some evolutionary traits that could help you to understand how this process have evolved. So that's my comment and question.
Okay, so there's quite a lot there to unpack. So to deal with your second point first, yes, we make no claims that what we are examining relates directly to aging in higher eukaryotes. All we're trying to do is establish a rationale for why it might be there. One thing to, I guess, to point out is that this is in no way unique to what we're saying about yeast aging research, because yeast aging research for 25 years has been saying aging is all about these ribosomal DNA circles, and it's a slightly awkward problem that no one's ever seen them in any other aging organism. So whether what we're talking about extrapolates, whether it doesn't, we don't know. We make no claims to that at the moment. All we're trying to do is to prove this point that there could have been an evolutionary purpose, and that may well be something to do with food or adaptation, and that might give us links into regulators. But that really, it's just a hypothesis generation exercise. In terms of your first question about the epigenetic memory, we have wondered about this a lot, because there has been some very nice studies of this. And our first set of experiments when we went into this was thinking, right, we're going to go in and we're going to knock out all sorts of epigenetic modifiers, and that will do something to aging. And it really didn't. So things that should have affected the memory. So this is normally conveyed by the various histone methylation marks, depending on which of the studies you look at. This is K4 or K36 or K79 methylation. None of them do anything really in the aging process. We just haven't found anything convincing. And also in terms of the memory, it doesn't matter whether the cells have seen galactose or glucose before. We actually try to make sure that they haven't experienced any shift at any time in the memory of the cells which are actually going into the experiment. So we try to make sure that everything that goes into the experiment has only seen the carbon source it starts in. So we don't think it's about a memory process. But the memory thing, it is supposed to be all about pre-adapting cells to environmental changes. So I'm not going to rule out that they're linked either.
Okay, thank you.
Thanks, Diego and Jesus.
Thank you. Very nice work. I would like to know what happens in your first part experiment, in which you change from glucose to galactose and raffinose. If you go into a real non-fermentative carbon source, a respiratory substrates like ethanol, lactic acid, glycerol.
Yeah, we did try this, and the cells just grew somewhat randomly. It's something we need to go back and re-examine, because to be honest, we've sort of retrofitted the ideas about what it's important for onto the experiment. And now we've got that, we need to go back and look at that again.
Thank you.
Hi, very, very interesting talk, very provocative. I have two questions. One is, if mutants that have defects in carbon catabolite repression show any defects in aging, because what you see is that the cells, as they age, they can now use different carbon sources, and it seems that glucose is not acting as a repressor anymore.
Yeah, so we did look at this. We looked at the MIG1 deletion. So that should firmly knock out the carbon catabolite repression. We didn't do this in terms of the competition assays, but we did look at this in gene expression because it was a fairly obvious hypothesis that MIG1 activity could be changing. But actually, either on glucose or galactose, we didn't see a change in what happens in aging when we deleted MIG1. So obviously, it does have gene expressions effects in young cells on glucose, but then that whole sort of genome-wide transcriptional derepression, that still happens. That's not driven by MIG1.
Okay. And my second question is related with the formation of the extracellular DNA molecules. I don't know exactly how the mechanism is, but there seems to be some relationship with transcription, like genes that are being transcribed more actively will generate the extra chromosomal DNA molecules. Is that the case?
Yes, it is. It is so the more we have dug into this, the more horrifically complicated we have found it to be. Because I'm also generalizing a bit, because actually two things happen. One is that you get a copy number change on chromosomes, and the other is that you get a copy number change on circles. We've got a big project that it's pre-printed at the moment. We're trying to get published, where we've tried to work out why transcription drives the copy number change on chromosomes. And the answer, as far as we can tell, is that when the gene is turned on, it recruits a complex called mediator, which recruits a complex called saga, which associates it with the nuclear pore complex. It's a process called gene gating, and this has been seen before. And when the cell replication fork approaches that constraint, which is caused by the link between the gene and the nuclear pore complex, it causes stalling of the replication fork, and we can see that as well. And what we think happens is at that point, the forks reverse, so you get these four pronged sort of chicken foot structures, which basically lead to the extrusion of a bit of DNA, which can undergo homologous recombination reactions and form circles. And again, we have all the genetic dependencies of those forming circles. And without actually making a matching deletion in the chromosomal DNA. So yeah, it's complicated. There's in our PLOS biology paper on the circles, we described that mechanism, and then in the bioarchive paper, we describe how the replication impediment arises. And at some point, we need to stitch those two together for the circles. We haven't done it yet.
Okay, thank you very much.
Gracias. Thank you very much. Very interesting, your talk, and very original ideas. My question was also related with the mechanism of formation of the circular DNA. And but now that you explained that it will be related to the replication fork, I don't know, because so what the question is if it is known in yeast that during active transcription there is DNA double strand breaks that allows the entrance of the transcriptional machinery. Could that happen? It has been shown, at least in mammals, in neurons, that in very active genes, there has to be this double strand breaks. So I was thinking that perhaps the same could happen in yeast. And so this is one question. And the second is that at the beginning in the introduction, you mentioned, like in your very first or second slide, that that you had a model of replicative aging, so that you will have the cells, that the mother cell that will still be alive but will no longer divide. So do you think that these post-mitotic cells, this will be more similar to the replicative senescence that occurs in mammals, for example? Do you think that they could contribute also to the wellness of the population, thinking about the evolutionary origin or maintenance of this phenotype? Perhaps it has also contributes benefits the fitness, not thinking of individual cells, but thinking in the population.
So to address the second part first, yes, we wondered about this as well. So these mother cells which have gone through their replicative lifespan, they are really badly characterized. So when people report on this, when they look at, for example, microfluidics, where you can actually visually follow cells up to their replicative lifespan, they always stop the reporting at the point where they stop dividing. And I've spoken to Gilles Charvin about this, and he was saying that what happens after that is sort of random. Sometimes the cells die, sometimes they don't. It's tell. In our system, it's different. So we're growing the cells basically in a shaken batch culture. So possibly probably no more physiologically relevant than a microfluidic chip. Certainly nothing like things are happening in a colony. What we find is that those mother cells seem to last for a very long time, because we have certain mutants which we know they're dead by, or they've hit their replicative lifespan by 24 hours, yet they are still fully there and by 48 hours, and then they still have intact RNA, because obviously we do the transcriptome, so we run the RNA out in the gel every time we do it. So we know that they're not sort of breaking up, and similarly their genome is fine, and they're not apoptosing. Could those be contributing to the health of a colony by some sort of synthesizing metabolites or whatever? Your guess is as good as mine. We just have no way to get into this, and so we addressed the question that we felt we could answer.
Okay.
Going back to your first question about the double strand breaks and the transcription, we have recently developed this method called TrailSeq, which allows us to do genome-wide double strand break mapping, because we thought this is exactly what we want to see. We thought when we turn on the genes, there's going to be double strand breaks forming, and if we say delete RAD52, so basically we stop the double strand breaks being repaired, we should see those accumulating. And we cannot see diddly squat. We have looked so hard at this, and there's just nothing there. Something that we've come to think about is we're not so confident that double strand breaks are always where other people say they are either, because it is possible in replication, by the reversing of a replication fork, to get something which looks like a double strand break, because it's got a single-ended break, which is not paired to anything. So that is by many definitions a double strand break, but it doesn't need to be repaired, because as the Houseley junction migrates, it just disappears again. It turns back into a normal replication intermediate. And we think when people do TrailSeq, but also NSeq and BLESS and all these methods, actually most of what is detected is those reversed replication forks. And we have not come up with a good way to actually distinguish them using these methods.
So, but then in post-mitotic phase, it could be, it is, you can interpret without this difficulty of the replication fork, right?
Mm-hmm. Yeah, so we've actually done trail-seq on aged RAD52 mutants, and there is we cannot see any evidence of double-strand breaks anywhere.
Okay.
They could be scattered through the genome and we'd never see them, but there's certainly no sites at which they're concentrated. Again, we were very disappointed by that because we thought there would be something there, and there's just nothing.
Okay, thank you very much.
Gabriel, you wanted to ask again?
Yeah. I was thinking about this idea that you were mentioning, that the things that you learn early in life later will be beneficial for survival, no? Like glucose, and you learn that this will be deprived later on. But then there is this also the idea that during aging, the things that you learn early in life later on will become detrimental. And so thinking in this second way, what you show is that this is beneficial, at least for this fitting experiment that you perform. But what I was thinking, considering the mitochondrial experiments that you run, is if you have seen whether these age cells will be defective in autophagy. Autophagy, as you know, it's associated with well aging. And so that's basically the question.
We haven't looked at it very closely, I've got to say. We have thought about it a bit. We've tried to put a couple of autophagy mutants in. It hasn't really worked because one thing we're restricted by is that any mutation that causes a serious growth defect, we can't analyze.
Okay.
And in terms of actually putting in the autophagy reporters, we put in the GFP ATG8 reporter, and we couldn't see anything by the imaging flow cytometry, which is sort of our standard assay. But imaging flow cytometry is not wonderfully sensitive. So I would say that we have made a somewhat half-assed attempt to get an answer to that, but we didn't get anywhere fast, and we moved on. We need to go back to it.
Thanks.
Anyone else wants to ask anything? No? If not, then thank you very much, Jan. It was nice to have you. Very interesting talk.
Thank you very much for your attention. And yeah, it was a pleasure. Bye-bye.
Thank you very much.
Thanks. Have a nice weekend.
Gracias, Paula. Hasta luego.